Inglês, porque é universal. Good morning to all. Welcome to this second session of the first series of AquaInvert webinars. For those that have been with us on this first session, already know that AquaInvert project is an European interregional project dedicated to the research and development of the production of some marine invertebrate species with commercial interest. The target species are the sea urchins, the abalone, the limpets, and eventually the sea cucumber, the detritivore species relevant for the multi-trophic integrated approach. Of course, that the project has more components and civil activities, but we are here today to speak about the reproduction and the early stages and life cycle of species that I have already mentioned. So our first speaker will be Ricardo José, which has a master's degree with specialization on aquaculture, and is working in Madeira at the Mariculture Center of Calheta. So I'm giving the word to Ricardo. Ricardo, please.
Thank you, Eduardo. I'm like Eduardo said, I'm at Calheta. I'm currently working with sea urchins as well as with limpets, and my presentation will be like the previous one about sea urchins. Let me just share my screen with sea urchins, and it's mostly a review of the literature. Like the title in itself says, I will be talking about life cycle and aquaculture. The first question is, why cultivate sea urchins? These are the main three reasons: stock enhancement, natural stock depletion, and market demand. Aquaculture for sea urchins is important, mostly because of these three. These sea urchins are consumed due to its roe. So in the first part, people collect them from the sea, from the wild, which leads to stock depletion. Then we can cultivate them and use them to stock enhance them, to do some stock enhancement even in aquaculture, as well as to provide sea urchins for the market. Regarding sea urchins, the gametes are easy to obtain. As we can see in this picture, we have a male and a female, as well as a process to collect them. The sea urchins, the gametes, can be achieved by injection of KCl in a proportion of 0.5 to 1 molar of chloride potassium in a percentage of 40 microliters per gram of body weight due to stress or vigorous shaking. We have tried these three methods here. The KCl has a high mortality rate. Stress was mostly because when we transport them from wild and in the buckets, they would sometimes spawn in high densities, as well as due to unfortunate circumstances when we had some problems with the sea urchins in the tanks, we noticed that before they died, they would spawn. Vigorous shaking is a method used here based on a visit that one of our colleagues did previously to Ireland, in which they would shake the sea urchins to obtain gametes. These methods are favorite because the mortality rate is very low. In all of the methods, we must wait around 30 minutes to have some gametes on the sea urchins, as well as to collect them. The collection is like on the picture on the right, we can collect them straight to seawater and then do the mixture of of, of the male sperm and the female gametes, and then we will provide them to put them on to develop. This is a very short resume of the life cycle of the sea urchin. We have an adult that will release gametes that will develop, that will be fertilized, and within 48 hours we will have a Chenopletes larvae. This will develop in 4 to 5 weeks in a rudimentary to a small sea urchin that will settle and will take some time to achieve adulthood. Here we can find um, a more detailed version of the life cycle where we can see the fertilization, the cell division, larval, the embryological development, larvae with 4, 6, and 8 arms. The first, when they achieve competence, meaning that when the arms are reabsorbed and transformed, and before the larvae becomes a small sea urchin by metamorphosis, we'll have also a post larvae that's the small sea urchin that will be transformed into an adult later on. The the next slide is also one of the early development, but this is in a more detailed scale. Here, then, larval as conditions for larval rearing. We have a density of 4 larvae per liter. They can be fed with microalgae. Here we have 2 of the most common larvae used in sea urchin rearing, and light should be provided for 24 hours. Into settlement stage, we have usually raceways with a low water column, aeration, low water exchange so that the larvae stand in the tank. They are fed in this when they are in settlement with natural biofuel. That's meaning that the tanks are prepared previously, and larvae and the sea urchins are placed in the tank. There was also some trials with microencapsulated shrimp diets from INVE. The results were not that good, so they say it's doable to use the microencapsulated shrimp feed, but the results are not high. Then they can be placed in an integrated multi-trophic system to provide sound. After this, I think the major bottlenecks that are seen in sea urchin larvae is not to achieve gametes, but the settlement stage in which the mortality rate is very high. And that's it. Thank you so much.
Thank you, Ricardo. Thank you, Ricardo, for your presentation. We have been following on the other session, and we'll follow on this the same system. Then the questions can be put at the end of the tangent of all the interventions. So please, at the end, write or ask whatever you want to need to know about the interventions that we are going to have here. Right now, it's time for Gercende Courtois, who is working in University of Las Palmas. She's an aquaculturist by nature, by heart, by formation for a long time. she is really the coordinator of this project, and as you, she has a lot of work on Haliotis and on macroalgae, and she's going to speak now about Haliotis tuberculata, about the reproduction and early life stages. So please, Gercende, it's your turn.
Thank you, Eduardo. Okay, I'm going to share my screen. Can you tell me if you see it in full screen? Yep, okay.
Yes.
Okay, so Haliotis tuberculata, it's an abalone species that is present in Europe, in the Macaronesian region between Azores, Madeira, and Canaria. We have a subspecies which is called Haliotis tuberculata coccinea, and we've been all working on its reproduction for various years. So today we're going to look at its reproduction and early life stages. So like we know, it's a gastropod mollusk, it's herbivorous. We have abalone species that are all around the world in different temperatures and adapted to the different condition. They consume benthic diatoms in their early life stages and know naturally macroalgae in the later stages. We differentiate the males from the female by the color of their gonads, and this color varies according to species around the globe. So people would think, why abalone? Why people would like to produce abalone? Because it's a highly valued seafood, especially in Asia, but now in Europe, people are interested about it, and especially the chef from the different gourmet restaurants are really interested by the product. The interest also of the product is that it can be sold in various forms. It can be sold fresh, live, frozen, canned, dehydrated. There are also some places where they are doing prepared food that include abalone. So here we have an overview of the different species around the world and the different aspects they can have. So here are species, Haliotis tuberculata coccinea in the Macaronesian region. Okay, so how does the abalone production cycle takes place? So the males and females are separated. They are born male and they die male, and they are born female and they die female. There is no sex change throughout their life. And the males and female will liberate their gametes in the water columns, where the fertilization will take place in the water column. They will develop into trochophore larvae, up to veliger larvae, and then finally they will lose their velum when they will settle and perform their metamorphosis. And there they will start their benthic life up to their adult stages. Therefore, there is a complexity in the production of the abalone because of these two phases, because the first phase of the production is pelagic, during the eggs and larval stages, while the second stage, from the post-larval stage to juveniles up to adults, the animals are benthic. And then different adaptations have to be done in the production cycle, taking this into account. So regarding the broodstock conditioning, how are people doing it? Well, around the globe, there are various options, from tanks being 30 liters in volume up to 3,000 liters. Tanks can be round, rectangular, square, all sorts of possibilities are seen around the world. Some people, depending on the temperature they have in the region where they are producing the abalone, are using recirculation to maintain their broodstock, especially to maintain the temperature, to maintain them at a constant temperature, while other regions are maintaining their broodstock in flow-through systems. For the broodstock, it's important to have good water quality, and to have the animals separated from their feces. What are people feeding their broodstock? In some regions, they are feeding them macroalgae that are collected from the wild or that can be produced. Others prefer to feed them compound feed, as that's a way to control the feed they are ingesting, and therefore to control the gonad development. And normally they are kept under natural photoperiod or under reduced light condition, as abalone are animals that tend not to really like light and to hide from the light. So here are some examples of the feed that can be fed to the broodstock, and then that are also being fed to the juveniles, with a variety of compound feed that are existing for the different stages, and examples of macroalgae. In that case, these ones are being produced in IMTA system in order to feed the broodstock and the juveniles and the adults. Now, I mean, it's important to also look at the genetic programs in order to produce the abalone and in order to maintain genetic diversity. Therefore, for these genetic program, it's important to be able to tag your broodstock in order to know where your juveniles are, what is the origin of your juveniles for the further crossing of for future reproduction. Therefore, the tagging can be done with some plastic tags that are being directly glued on the shell of the animals. Others are using some pit tags in order to mark the animal and to be able to find them easily when they are selecting them for spawning events. There's different marking techniques for the broodstocks. So here I'm going to show you a small video of our abalones. No, this one is not working. Okay, so this is just to illustrate the maturity level of the broodstock and how they are selected according to their gonad index, and to have a visual of what we consider a gonad index of 1, 2, and 3. So different methods that are being used in order to induce spawning. The first one that can be used is the thermal shock, where the water temperature in males and female tanks is rapidly increased by 3 to 6 degrees, and then gradually decreased until reaching ambient temperature. This method can easily be done, but in order to obtain spawning by thermal shock, the animals need to be really mature. Animals that won't be really mature will not react as much to thermal shock. Another option for spawning induction is a UV radiation by passing the water through a UV lamp. And it's highly used in Japan, in Australia. The disadvantages is that the high initial cost for the installation of the UV lamp, but the benefits is that it enables to obtain high gamete quality, even in animals that are not fully with the animals not needing such a really high gonad index in order to be induced. The third option is the use of hydrogen peroxide. So normally in that case, first the animals are placed in water. In this water, there is a buffer that is added to control the pH. Normally it's tris, hydroxymethylamine. And then the hydrogen peroxide is added. The disadvantages is that the chemical reagents can harm the gametes. And then if the gametes get in contact with them, then they cannot be used to be fertilized. Therefore, once the animals are induced to spawn, they need to be washed several times in order to be placed in clear seawater in order to be able to use these gametes. And the benefits is that it's low-cost techniques, and that these reagents can be easily stored and maintained for a long time. So if we go to spawning and fertilization, normally the males and the females are induced separately, and then the gametes are being collected and then maintained in contacts in a bucket during 30 minutes for the fertilization to take place. So here on the top, we see female gametes, and here we see the expulsion of male gametes. The female gametes are sinking, and therefore, in order to be collected, they are then being siphoned from the bottom of the tank. When the gametes are siphoned, they are passed through around 400 micron screen sieve in order to eliminate the impurities that can be siphoned from the bottom of the tank. And then once the gametes, the male gametes are going to be in contact with the female gametes during 30 minutes for the fertilization, they will then be sieved through a 63 micron mesh in order to remove the excess sperm that would be around the egg and that can cause polyspermia. After 1 hour, the buckets are sampled, and then this enables to estimate the fertilization rate, where the eggs that are showing the first division are considered as fertilized, whereas the ones that are not showing the division are considered as unfertilized, and there, as a consequence, the fertilization rate is being calculated. So here we can see the expulsion of female gametes in a tank. So you see that the gametes are being expelled from the pores, and that they will slowly sink to the bottom of the tank, where we will be able to siphon them in order to perform the fertilization, to place them in contact with the male gametes. Here, normally once the fertilization takes place, they remain in a static condition until they start to hatch. When they hatch, they will become veliger larvae, and here you can see how they are behaving once they have hatched. You can see the the little spirals of larvae that are taking place in the bucket. And these ones will be the larvae that will be transferred to the larval tanks. Okay, so when we get to larval tanks, larval culture, there are two different ways to produce them. Some people like to have the larval culture in static condition, which means that this requires partial or full water changes every 24 hours, during which the larvae are being sieved through normally 63 micron mesh, and then replaced in the larval tanks that will have the water being renewed. Other people prefer to have open flow larval tanks in which the sieve is placed within the larval tank, allowing to retain the larvae and have a constant flow of water in the tank. In our case, we decided to go for the open flow system. So here are the larval tanks that include the sieve, and then the water is filtered to 1 micron and UV sterilized during the larval culture period. When we started with the Haliotis tuberculata coccinea, we had no idea of what was the timing of its embryonic and larval development. So we decided to go through all the development and estimated at what time the different stages were appearing. For this, the experiment were done with larval culture at between 1 and 5 larvae per milliliter. We've observed 39 different stages from fertilization until the appearance of the third tubule on the cephalic tentacles that indicate that they are ready to be transferred to the settlement tanks. Here you can see the diameters of the eggs and then the larvae once they are prepared for metamorphosis. Here are some veliger larvae. Okay, sorry. Next one. Okay, so therefore the critical stages in the production is the settlement and metamorphosis. If the substrate that the larvae will encounter is not suitable for the larvae, we can get up to 99% mortality. Therefore, there is a need to adapt the quality and the nature of the settlement substrate to the post-larval requirement. So the abalone larvae for the different species have been tried to be induced to settle and metamorphose through the use of gamma-aminobutyric acid, the use of congener mucus, Christos coralline algae, uvula, different diatoms. And according to the use of these different substrate, your settlement rate can go from 1% or less up to 40, 60, and in some cases over 60%. So here we've decided to go for, our settlement tanks are of 2,500 liters. The inside we've placed some polycarbonate plates that are the ones that are being covered by the different substrate that we are using. They are kept under natural light and temperature, and the water that reaches the tank is filtered up to 1 micron. Here are some examples of settlement and metamorphosis in Australia, in the Philippines. And here we observed a man feeding the post larvae once settled with diatom that is going to be their primary source of nutrition. Here we observe other settlement methods that is in Thailand, where instead of using fixed and hard settlement plates, they are using some plastic sheets in order to settle the post larvae. Out of the different experiments that have been performed, we've observed higher settlement on Christos coralline algae, like it's been observed for other species of abalone. In our case, the experiment we've done with gamma-aminobutyric acid, mucus, and diatoms did not give some interesting results and were not significantly different from the negative control. On the other hand, we've tried the use of Ulvella lens, and we've observed that its efficiency varies according to the age of the Ulvella lens, the enrichment method of the Ulvella, and if it's been covered with diatoms or if it's not covered by diatoms at the moment of the settlement. And so people would ask, well, why are you not using Christos coralline algae if it works great? First of all, covering the plates with Christos coralline algae is quite long. So if you need to have your settlement plant plates to be regularly being covered with the substrate, this is not really practical. And then another thing is that the Christos coralline algae have a natural systems where they are peeling in order not to get colonized by other algae. And therefore, this prevents the diatoms from growing on top of the Christos coralline algae. So they're great. You're going to have a lot of settlements on your Christos coralline algae, but then they will not be able to feed because the diatoms will not be able to remain on the Christos coralline algae. Therefore, we've decided to go for the Ulvella lens, which gives a bit lower settlement amount, but then in terms of growth, it's more interesting. And then thank you very much for listening. And then in the next webinar, we'll look at the nutrition of these early stages and up to the juvenile stages. Thank you. I will stop sharing.
Thank you, Gercende. It was a very nice presentation. What's happening?
I don't know.
Okay. Well documented. I think that it was very interesting, at least for me. I think that the others will also enjoy very much. And now we are going to Diego Castejón, a postdoc working at Madeira with Limpets, who is going to talk a little bit about the early approach in the production and culture of limpets. Please, Diego, it's your turn and it's your field.
And thank you very much, Eduardo. Now we'll see here, starting here. Well, good morning to everyone, and thank you very much. Since the Limpets culture is still experimental, many of those data is still in develop. So today only will be see an early approach about the production and culture of Limpets larvae and juveniles. First, we will start with a small bit regarding the identification of the species and the sex of the animals, and then we will see step by step and all the required for the juvenile production, starting from the obtaining of the gametes, larval production, larval culture, and finally juvenile production. We have two limpet species, Patella aspera and Candei. Major difference in regard to the body color, typically yellow-orange in aspera and gray in Candei. the mantle tentacles that tend to be more visible in Aspera, cell thickness that is thicker in Aspera, and the color of the female gonad, as we will see later. The reproduction of fertile individuals in both species usually appear between October and March. Limpets, are different from abalone, has no external sexual dimorphisms. So if you would like to check the sex of your specimens alive, you need to realize a biopsy. Here you can see the colored chocolate that characterizes a female Candei, the red color of the female Aspera, and a white or cream that is regarding for the male of both species. Limpets also are totally hermaphrodites. This means the first maturation occurs as a male, and later on, in a later age, the animal maturates as female. So you need to consider this because usually higher specimens to be females. So the animal size will have influenced your adult stock and yet the production of gametes. For now, two methods are possible to obtain limpets in gametes. To obtain gametes in limpets. The first one is the spawning induction. This method ensures the survival of the adults. Usually the quality of the gametes is high, but this methodology is still experimental in limpets and didn't show a very good reliability to know. On the other hand, we have the dissection, the fertilization in vitro. This method the adults cannot survive this methodology because you need to dissect them, and the quality of the gametes vary from very low to very high. But this methodology is developed and tends to show a very high reliability to both the obtaining of the gametes as well as the production of larvae. Here you can see the dissected specimen showing the gonads. You only need to cut the mantle and obtune the food. That is this structure here. And in Patella candei, the females have had a gonad called chocolate. In Aspera, the gonad is colored red, while in the males of other species, the gonad varies between green and white. To obtain the sperm, you can collect the sperm directly from the gonad using a plastic pastel pipette. Then the sperm should be diluted in for example, in a beaker with clean water. The dilution of the sperm in the water is enough to activate the sperm. To maintain cleaner condition, it is recommended to filter the sperm solution to remove the debris. And finally, I also recommend to pull the sperm of several specimens. Just in case of one specimen or more had a sperm with a low quality. For the oocyte, we place the entire gonad inside a beaker, and using the suction force of a plastic pastel pipette, we broke the gonad and release the oocyte. We will obtain a mixture solution of oocytes and debris. So, at continuation, you will need to filter the solution. A first sieve to around 200 microns to trap the debris, and a second one with around 50 microns to save and wash the oocytes. Then you need to place the oocyte in clean filtered seawater. Here in this picture, in the left, we had a pool of oocyte of Candei, and in the right, a pool of oocytes of Aspera. But the oocyte just extracted from the females are not ready for fertilization. You need mature oocytes. And the process of maturation can be realized artificially using alkaline seawater baths. This step is tricky. Too much pH or too much time, and you will obtain deformed oocyte, just like here. These oocytes are not viable for fertilization. So you need to consider the time and pH for a good or optimal maturation. After several experiments, we found that using sodium hydroxide as alkaline agent, for Patella aspera, the pH more recommended looks like around 9. In Candei, it's around 9.5, and the duration of the bath is 3 hours. After the bath, you need to clean and wash very well the oocyte. Then, with the maturate oocyte in clean seawater, we can realize a static fertilization in which we place the sperm with the oocyte during additional 24 hours, that is the typical time for incubation. And we can maintain the sperm with the oocyte without any kind of problem. With this method, we had a larva production between the 50 and the 75%. In other experiment, we also tested the role of the ammonium hydroxide. With this compound, the maximum larva production can be obtained in only 10 minutes, so we can save a lot of time. But the problem with ammonium is that it can be very reactive. So if you are not careful, you will obtain a high proportion of deformed oocyte. So depending on your timing, your organization, and your requirement, you can select one method of another for a short bath or a long bath. In other set of experiments, we check the sperm concentration. And the optimal one in Candida and Aspera looks like around 100,000 sperm per milliliters. A low quantity of sperm, and we will obtain a lower production of larvae and a higher density of sperm, and we obtain a lower production because polyspermy. So sperm concentration should we consider. In parallel with the larva production experiment, we also check embryonic development. And in Limpets, it is very fast. In less than 24 hours, we can obtain Trochophora larvae. Trochophora is a free swimming larval stage. It's planktonic, it's very mobile, and it's characterized by a crown of cilia as well as apical cilia that we will see in a more here, that the larvae used to swim. Trochophora can be found usually in the upper layer of the water column. And with the Trochophora, we can start the larval culture. We first need to collect the Trochophora. We can siphon the larvae and gather them using a 50-micron mesh. Then the collected larvae can be placed in beakers with clean water and a low density, around 10 larvae milliliters. These conditions are more than enough than in 48 additional hours, 2 days, obtain pediveliger larvae, the last larval stage. These larvae and feeding is not required because larvae are less heterotrophic. This means that all the energy that the larvae needs is obtained from the yolk reserved from the oocytes. Only one important thing here: never ever collect the material from the bottom on the beaker, only the materials from the water column, because we found this. And the bottom of the beakers usually contain non-viable specimen, debris, decomposed matter, artificial fiber, and even worse, larvae aggregation. In my opinion, the 90% of these larvae are viable, but they are aggregated themselves. And this is still a problem that we can solve. We expect in the future don't have this kind of problem for now. But for now, never collect material from the bottom, only the water column. Here we have the pediveliger larvae. It's the last larval stage, only 3 days post-fecundation. The pediveliger is characterized by the protocols. It is the larval cell, velum, the organ that the larvae use to swim, eyes, separate tentacles, and the foot that the larvae can use to grow. We use this stage only 3 days after the fecundation to start the settlement test. Pediveliger can swim, is the most typical locomotion behavior of this animal, and also the pediveliger can crawl. Sometimes the crawling behavior is used to decide the larvae is in the bottom, it's settled, but no, it is false. A crawling larvae can start swimming in any moment. While realizing our observation and monitoring of the larvae, we found crawling larvae starting to swim because crawling larvae in this moment is checking the substrate. If the substrate is good, the larvae can settle. If not, the larvae will start swimming again. We realized a settlement test using culture cell plates, in which we can test different kinds of substrates simultaneously. Here we have a control with nothing but water, a plate, a dual cell with coralline algae, different diatoms like Navicula or Pharamphora, a blob because it's usually recommended for the culture of marine gastropod, and here we have a mixture of diatom plus blob. And for this element, we identify 3 different stages. For comparison, the first one is the periveligia. We identify 3 different stages. For comparison, the larval stage is not celled. There it had a verum, like a band around the face that you can see through the cones. It's not able to raise and only had the protocones, that is the larval cell. The next stage is the early post-larvae. It's a post-larval stage. It's for first time settled. The verum disappears, so cannot swim, but is still not able to raise and only had the protocones. The next stage is the juvenile. The juvenile is able to raise and develop the teleocones of the juvenile cell. In our study, to consider this element, we only consider this stage and this one, and nothing more. Here you can see the post-larvae, they didn't move very fast, tend to stay quiet, while the juveniles didn't stop. They are continuously moving, continuously grazing. Only rarely we see a stopped juvenile. Regarding the experiments, the most important result can say, in Patella aspera, the only substrate that induced the cell element were clusters coralline algae, while in Candei, only the corallines and the diatom biofilms of Navicula induced the greater ratio of juveniles. So it is a very interesting result because Navicula is very easy to culture in comparison with the corallines. Also, in both species, Aspera and Candei, the corallines induce the faster cell response, around between 2 and 3 days before control. And also, corallines are not an unitary things. The corallines form different kind of community without the corallines and other macroalgae. So we found that different kinds of clusters coralline algae communities had differential influence of the larval cell element. So if you need to use coralline for any case, be careful for what you are selecting. The corallines have some advantage because they are very easy to obtain. We obtain the corallines directly from the limpet cells. They can promote massive cell element. Here with that test, we realize it with an entire cell, and each yellow dot is a post-larvae, so the response in some cases can be very good. And the juveniles also can grow the corallines. They can feed, the juveniles can feed with the substrate and grow. But it's not everything is happy. The corallines had several problems to solve. For example, the corallines are complex communities and include some inductors as well inhibitors for cell element. The corallines can degrade over time. And if this thing happens, you will have a high larval mortality. So the research of the use of the cluster coralline algae is still under research. To finish the presentation, you can see that the cluster coralline algae can be useful as a food source. Just tell before, the corallines can set layers of cells, but in my opinion, maybe the limpets can use this layer of dead settings as a food source. And in fact, you can see that the cell of the juvenile is covered in remains of fishes made of corallines. And a better result, Navicula that give a good result in the settlement of Candei also can be useful as a food source. Here you can check the grazing trails. This means that the larvae has been feeding in the substrate, and the larvae can grow, increasing the size in short times. And here you can see a juvenile with feeding behavior. In conclusion, you can say that the larvae or limpets can be produced oocyte obtained by dissection, and mature artificially using alkaline seawater. The optimal bath requiring around pH 9.5 during 3 hours if using sodium hydroxide, or 10 minutes if using ammonium hydroxide. The larvae develop to the last variety stage in 3 days without feeding. Juveniles can be obtained using crustocalin algae, and aquaculture of Patella candei is promising because navicula induce this element and can be used as a food resource for the select juveniles. And the 2 last references about a study about Limpets production. And thank you very much.
Bravo, Diego.
Very well, Diego. Very interesting. I think that Mirko has a lot to talk with you after this presentation. But we are going to now to Mirko, who is a postdoc working here in the Azores, also in Limpets. He has been working for a long time with other organisms also, and he's going to speak about limpet settlement, of course, what we have been doing, and probably that's why I'm saying that it has a lot of things to discuss with you, Diego. Okay, Mirko, please.
Thank you, Eduardo, for the nice presentation. Yes, Diego, I have a lot of questions for you. Maybe we will have to change ideas, but also with Gercende, I have some question for her. Anyway, I will share my desk. Let me see if everything is fine. This is my presentation. Share. Are you seeing the presentation?
You need to change it. We are seeing the.
Yeah. Okay.
We are seeing the one with the.
Okay.
You need to change the screen.
The display.
Yeah. Okay. Now it's okay.
Yes.
Yes.
Okay. Now, Mirko.
Okay. Perfect. Okay. Well, as Eduardo told you, I'm Mirko de Girolamo, and today I will talk to you about the progress achieved in the framework of AquaInvert project regarding the settlement and spawning of the limpets in the Azores. First of all, I would like to tell you about the Aqualab, the place where all these research activities are carried out. Aqualab is a small facility of the Institute of Marine Research Oceanos of the University of the Azores. And since 2008, we have been carrying out researches related to spawning, larval development, and early growth of different species, such as the giant barnacle, that is Megabalanus azoricus, an endemic species of the Azores, Oluturi santori, the sea cucumber, widely distributed in the North Atlantic, but also the Mediterranean Sea, the European abalone, Haliotis tuberculata, and more recently the Azorian limpet and the black limpet. There are many reasons, because we are interested in these species, and one of them, of course, is the economic interest of these species, but also because these species are sensible to the, for many reasons, are sensible to the fishing. So one of the aim of the project, AquaInvert project, is to promote the sustainable aquaculture of these species, not only as a way to diversify the fisheries products, but also to relieve fishing pressure on these species. And in some local population, where these population are depleted, maybe also to use this restoration way to restore the local population. But first, we have to get there, and to get there, we need to control the reproduction. The first step is, as you saw before, for the limpets and for the abalone, is how to get the gametes. Mainly, there are currently there are three main methods that are considered. One is non-lethal biopsies. Other is the induction with different factors of release of sperm and eggs, and at the moment, the more reliable system is the dissection of the gonads. Whichever method is used, individuals must be mature, and the use of injectable synthetic analog of some gonadotropin releasing hormone could be the solution to improve the maturity of the gonad. This hormone has been often used in other hatchery for fish, and more recently with a certain success in Hawaiian Limpets, Lana sandwichanensis. The fertilization rates of the oocytes can be improved. Using, as you have seen before from the presentation of from Diego, can be improved using a treatment called activation method, which consists with increase the pH of the seawater. The increasing of pH induces an increase intracellular increase of pH, which ultimately unblock the meiosis arrested to the prophase stages. In this process, the external layer of the eggs is dissolved, and this is a signal, to understand if the eggs are activated or not. Of course, there are many alkalization agents, and that can be differently efficient. In our experiment, we try to understand the different efficiency of these agents, and using the same amount of using the eggs from the same spawning, and using the maturation with these agents, increasing the pH from pH to 9.1. That was an intermediate point between 9 and 0.5 that Diego showed you before. And we count, of course, the eggs which were without or partial eggs. We did this on Patella aspera and Patella candae. Of course, the maturation time, the maturation of the eggs increase with the time, and again, after 3 hours, more than 80% of the eggs were activated. The same percentage, we get the same percentage after 1 hour and a half, using ammonia hydroxide and ammonia chloride. Of course, the behavior, on average, the behavior was the same both in patella candidate and aspera. But what we didn't know, and Diego explained very well this fact, what we didn't know, which was the effect on later development. And this is something that we want to explore in the next future. Anyway, the gametes can be obtained also by induction method, and the induction method uses different factors that can be, generally speaking, divided in physical and chemical factor. Physical factors are supposed to simulate the natural condition, like thermal shock or bubbling, simulating the wave action, or dry and wet condition, simulating the high and low tide, while chemical factors are instead endogenous factors that like UV light and hydrogen peroxide, which promote, which produce free exposed acts, because they produce a free radical, which ultimately promotes the production of precursor prostaglandins, which are well known, which are important, which have an important role in the reproduction of mollusks in general. So what we did was using the same number of patella, in this case patella candidae, but also in patella aspera, we do the same number of individuals divided by sex, and we try to induce the emission of sperms by a strong flux of air through the thermal shock, increasing the temperature for more than 3 degrees Celsius, using low percentage of hydrogen peroxide, and using UV light with a power of 500 milliwatt-hours per liter. We were really lucky, or did well our job, because we get gametes with both air, with the bubbling temperature, and hydrogen peroxide. Of course, the numbers are still low, but we are confident that with this one of these methods, probably the bubbling, we will get the induction of the gametes. Unfortunately, we didn't get gametes with UV light, but we will work on this treatment also, maybe increasing the power of the UV light. Once obtained the gametes through these two methods, induction and dissection of the gonads, we have been able to compare data from the induction and dissection, maintaining similar conditions, of fertilization, like concentration of gametes, temperature, contact times of the gametes. We get after 3 hours, we count number of the percentage of fertilization, and as you can see, we get a higher number in the induction compared with the dissection. Then we transfer healthy trochophora in a static vessel, using again more or less the same density, and after 72 hours, we again check the number of competent larvae, and it was almost 100% in the induction method, and more or less the half in the dissection method. Again, we use the healthy these larvae, the competent larvae, for a settlement test, using a flow-through system with the same plate, with the same biofilm, and after 1 month, 30 days, we check the survivorship, which was low, but still higher for the induction method, section method. So was evidence the advantage of, even if this preliminary test were evidence the advantages of the induction method, which is the eggs are ready to be fertilized, are active and ready to be fertilized, and of course we get better survival and better settlement. Still, consider that at the moment the section is still the most reliable method. Well, once we get after the fertilization and after the early development, the settlement process start, and this process is a transition from a planktonic larvae to a benthic juvenile. During the early, after 48 hours, early valiger differentiating to a post-torsional valiger, going through the turn, the clockwise turn, which is typical of the limpets, and the mollusc, going through the clockwise turn, which is the turn of visceral portion relative to the head portion. After 72 hours, this post-torsional valiger differentiates into pedivaliger, which maintain several larval and adult structure. At this stage, the larvae are able not only to swim, but also, as you've seen before, are able to crawl on the substrate. And this is the moment in which the larvae explore different substrates. If the cues are right, after hours or days, they go through a metamorphosis, and in which in this transition, the larvae go through the reduction of the verum, and they are called at this stage post-larvae. So it's important to understand which are the cues for the settlement. This is a key moment in the management of the life cycle of these species. Of course, we don't have any, not any, but the only cues we have is to refer to previous work on other gastropods, and especially to the abalone. So, as you have seen in the previous experiment, one of the first cues to be tested is the diatoms. Diatoms are microalgae that can be benthic or pelagic. Of course, the primary source of food will be the benthic diatoms, which, by the way, are also found in the gut contents of juveniles and adult of limpets. another good candidates, and now you see from the previous experiment that Diego showed you, another good candidate is the coralline crustacea algae, which are often associated with the settlements in Abalone. Recent work has also found a great association of these algae with the recruitment of a subspecies, Patella lysiponensis, in the coastline in main Portugal. Other tools can be the mucus released by the food of juveniles and adults. This can be a strong signal for the larvae to choose the place where to settle down. Again, Urvella lens, Urvella leptocete, Urvella are species that can be used for the settlement as a cube for the settlement of the larvae. The advantage of the Urvella is that this is a benthic macroalgae, the green macroalgae. Is crustus macroalgae growing as a flat rosette, and differently from the crustus coralline algae, is not calcareous, and so can be used for a long time by the larvae for the early growth. The same we can say also for the germs of other green algae, which are Ulva lactuca, Ulva rigida, Ulva clatrata, different species. It has been proved to these germs to improve the settlements in different species of abalone, and we believe that's also in case of limpet can be a good choice. So, trying to improve the settlement in a most rapid and extensive way, we conducted preliminary trials with Patella candae using different substrates. In this case, was Ketocerus, the atoms like Ketocerus and Amphora, Urvella lens and leptocete, and Ulva and the germs of Ulva lactuca. We also test different substrates, including volcanic stones, which we believe can be substrates which the larvae like. We use Pediverigers or Pediveriger competent larvae from the previous spawning induction, mainly the thermal shock and public method. Using 200 larvae per vessel with a system close and an exchange of fresh water of seawater every almost 2 days, maintaining the temperature of around 70-80 degrees. So what we count the Pediveriger. The Pediveriger on the substrate and on the different biofilm was Pediveriger were there until at least for 2 weeks exploring different substrate. There were almost no difference between substrate and biofilms in this stage. But at the settlements, we start to see our first settlers After 10 days, it means that with the right tools, the settlers can be settled down relatively in a few days. The settlers were present in the biofilm with uvella lens, both in the test of biofilm and of test of with substrate. And there was a clear preference for uvella lens, even if we saw some indication for the preference of the diatoms amphora and for the uvella leptocyte. Unfortunately, we didn't get so many settlers of the substrate, so we didn't conclude almost anything. But so to conclude, our next for the future of our research in the framework of AquaInvert project, we intend to, regarding the fertilization, we intend to optimize the methodologies to obtain the gametes, mainly the induction method, because we don't need to sacrifice the spawners. But also try to understand and improve the fertilization rates using different agents that can be a versatile method to get them to the fertilization. And of course, improve one of the key moments in the hatchery management, that is the settlement of the larvae using different tubes. With this, I conclude my presentation. Thank you for your attention, and I will stop to share my presentation now.
Thank you, Mirko. Ricardo, can you please?
Yeah.
Yes.
I'm here.
That's it. Thank you. Thank you, Mirko. As I can see, you and Diego really do have a lot of things to talk about and to discuss on the next meeting. However, we have a lot of questions here, and we have to go and follow them. I think that I'm just going to the beginning and doing it in a row. So the first questions were for Ricardo. Gercende asked if you can indicate what settlement indicers are used for the sea urchins. Ricardo, please.
Okay, I'm a bit confused with the question. What do you mean with settlement indicers, Gercende?
I think Diego had the same question. It means once you have the larvae and once they're ready to settle, what substrate are you using to settle the sea urchin larvae?
The literature what speaks is mostly natural biofilm. As long as you provide food and a stable temperature, the larvae tend to do the settlement process. Otherwise, if you provide some sort of control on these two parameters, we can have some delay in the settlement. Does that answer your question?
Yes, and the biofilms is normally a bacterial biofilm or diatom biofilm.
They say natural. They don't really explain, so I would assume probably a mixture of both, between diatoms and bacteria, and all of that.
And in your case, what are you using?
We are not at that stage. We are going to use natural biofilm.
Okay.
Yes, I think, Ricardo, that you have to understand what natural biofilm is, because it's quite variable, right? So I think that Mirko had exactly the same doubt, right?
Yeah, the question was almost the same. But if I can ask to Ricardo, do you think that maybe other inducer, other like Gercende, Ricardo, were talking about the coralline crustus algae can be a nice inductor or
coralline algae probably are a factor, especially because in nursing areas here in Madeira we find the sea urchins in tidal pools. Okay. So that's why we think that coralline algae will be useful as well for the settlement of sea urchins.
Yeah, perfect.
Diego was wanting to know, Ricardo, if the potassium chloride for the action was injected through the piercing or through natural.
No, what we do is we put the needle in a more or less 45 degrees angle and try to place it between the teeth and the membrane so that we don't harm the animal.
Is that clear for you, Diego?
Yes.
Yes? Okay.
Thank you.
You're welcome.
Now it's time for Mirko to answer. Next questions are for this person. No, Mirko is asking to Gercende. Now it's time of Gercende. Gercende, which are the advantages of comparing static and flow-through systems for hatching? Asks Mirko.
And Mirko, what do you mean that for hatching?
You were talking about you are using a flow-through system for hatching, but there are other systems like a closed system. So my question was, do you have an idea which are the advantages between the two methods? If it's just because you get better results.
The hatching, I mean, what you have is that you have you can more control your water if you want to control the temperature or if you want to be in stable condition. This is a way you have of controlling, but if you do it in closed system, that means that every 24 hours you have to do a water change manually, and you have to manipulate the larvae that have to go through the sieve. In our case, we thought that this was a lot of manipulation for the larvae, so that's why we decided to go for the flow through. But some people prefer to do it in closed system, so I guess it's up to the condition where you are. It depends. If you are in a place where you have to heat the water, of course you're going to go in a closed system because you don't want to go in a flow through. So it depends on your environment.
Yeah, so at the end is more a practical choice than there is no the advantage to choose one system compared to the other is just because it's more easy in a way for you to get the flow through. I'm asking because we are at the beginning, we were also trying to decide which system, and we try to test in different way if one system was better or the other, but at the end our decision was, as you were telling us, practical, because it's much more easy for us to have a system with the water going through, and that's it.
And you have another question, Mirko, about the germlings of vulva being used or not for settlement. Gercende, do you have any experience on this?
Yes, we've tried the germling of vulva in one of the experiments that we performed, the old germlings. When we say old, the time that we tested was maintaining them in culture for 45 days. They gave some interesting results. But from the experience we have also with the Ulvella lens, you realize that depending on how you're enriching the algae, the spores of the algae, and how long you are cultivating them on the substrate, will make a difference in the settlement inducing power that they have. So in other experiment, we've retried with ulva germlings, but the way of spawning and the way of enrichment was different, and they did not give such good results. So at the end, the way that you are enriching the algae in order to have them releasing the spores, and the time that you are having them on the substrate before you bring the larvae, does seem to have an impact, independently if it's the same species or no.
Okay. And do you think that could be a good idea that the production of germlings of the plates, because in this way maybe you get the standard method, because I thought that you get a lot of good results in some experiments with the germlings of ulva, and we did also. But of course, is really variable. So from a certain point of view, it's really good method, but on the other side, you don't get a lot of settlement there. I was thinking that maybe if we have a standardized method to produce germlings, we can have a good idea in which way the larvae behave for the settlement.
Yeah. I think that would be needed, because we need to make sure that when we talk about Ulvella lens, or when we talk about Ulvella or ulva, sometimes, I mean, it depends on some of the practical reasons. Sometimes you can leave them for 15 days, or sometimes you have more time and you can leave them for 3 weeks, or sometimes you don't have time and you have only 1 week to have the spores on the plate. So it's true that it's not standardized, and we would need to standardize the method and the enrichment also that we are putting in the tank in order to obtain the spores, in order to make sure that we all do it the same way, because otherwise it's hard to compare. When you tell me, oh, ulva is working for me, and I'm telling you, oh, ulva is not working for me, but did we induce the germlings the same way, with the same amount of nutrients, with the same amount of light, with all this will definitely have an effect on the induction of settlement capacity of the germlings.
Okay.
Thank you. And Carla also has a question for you, Gercende. How long do you usually, normally wait to fertilize the gametes after their release? And,
normally, we, as soon as the gametes are being released, we try to fertilize within 1 hour from the time the gametes are being released. We give us like a window of 1 hour to have the maximum fertilization rate. After that, you can still fertilize, but the fertilization rate is starting to reduce.
I think it was quite clear. Carla, I don't know if you want to assure us that it was very clear. Okay. Carla? Yes, okay.
Okay.
Another, Sylvia. Sylvia also wants to know, Gercende, how do you control Uvella growth because Sylvia has found that very dense uvula substrata apparently prejudices small juvenile urchin growth.
Okay, so
So
Sylvia, you've tried the uvula lens for sea urchin settlement? That's a question.
Yeah, we have uvula viridis actually. So we made a very simple experiment because we put the uvula spores in the aquarium, glass aquarium, and let them grow there. So, but then the uvula stay very thick, a very thick substrate, if you can say that, and then the urchins didn't grow. So. Yeah. I think we have here is a problem then when they settle, they are not really proper to eat macroalgae yet.
Yeah.
So I think that and after seeing your presentation, I was thinking about trying to grow uvula and diatoms at the same time, but then, well, for us it's like complicated because we don't have the right light conditions to grow algae in the lab. So okay. I'm struggling with that at the moment.
Would you have a place to have natural light outside of the lab, or no? You have no.
We could have it, yeah.
Because what we have observed on our side is that normally the uvula grows better, or release the germlings better when it's under natural light than when it's under neon light. And at the time so the thing is that the uvula releases the spores, and then of course, like you say, we want to control the development of uvula, because especially when we wanted to test it in older. When we wanted to grow it for a longer time, we had the same problem. If the uvula grows too much, then the post larvae cannot settle and they don't like it, and so then they kind of get entangled, and then they die.
What happened? Yeah.
So what we did is that we played with the amount of nutrients that we had in the tanks, and then we played with the light also. So we would put the nutrients and full light in order to release the germlings, and then leave them for a few days with this, and then we get rid of the nutrients from the system so that they don't grow as much, and then we tend to play with by covering them part of the day and getting the cover another part of the day in order to control the growth of the uvula. Without without it dying and having the minimum amount, but then that way we could have uvula in the same amount of cover, even at different ages, because also we had to have the same amount of cover in order to compare. Because if I have a 4-day-old uvula and I want to compare it with a 40-day-old uvula, but I have one that is covering 10% of my plate and the other one that is covering 90% of my plate, then I don't know if it's the effect of the uvula or the effect of the percentage cover of the uvula. So we played with this, and it was working quite well. And then if after a few days of getting rid of the nutrients, then we put some more nutrients in the system, and then we get rid of them, and then playing with this, we were able to control
Okay, good idea, nice idea, yeah. Yeah, thank you,
great.
Gercende.
And what else did you try for the settlement of sea urchin?
Well, we tried microalgae biofilm, but it was not well. We tried diatoms, but they were not venting diatoms, so we had troubles like controlling the quality of the biofilm. But now we have amphora, and we'll try with that. And also using like the glue of the sea urchins also, and also the waterborne of the broodstock. And well, but our problem now is like putting the urchins eating having the right substrate to they start eating when they settle, because there is a lack of time between settlement and they starting eating. So we are not able to control that yet.
Yeah. You can then try to cover the Ulvella with the diatoms. This works well for the abalone because at the beginning the radula is not sufficiently developed in order to eat the Ulvella, but then the diatom can cover the Ulvella, so they can start with the diatoms and then they can have access to the Ulvella once they are ready.
Maybe another solution can be germins of ulva.
Yeah, actually, yes, that's a good idea also, because they are supposedly, I think so, more like fragile, so they are more easy to bite, I think. So
Definitely.
Could be a nice idea also. Yeah, our problem here is the light to control the growth of the microalgae. It's difficult for us. Yeah, in the lab it's very difficult.
Yeah, but in the lab, at least to get the plates is not okay. Of course, you have to test your system, but it's something that you can get in a more or less simple way.
Yeah. So, thank you. Yeah.
Okay. Thank you, Sylvia.
Okay.
For this dialogue also.
Okay. Sorry for taking control of it.
It was a pleasure.
Okay.
Okay. Now the next question is from Mirko to Gercende. Theoretically, the best plate for settlement should be half CCA and half diatoms, right? Asks Mirko. It's more or less focused on this, what we have been discussing.
Yeah, of course. We are in the same ship.
Yeah, theoretically that would be ideal, but then the problem is, so that they have the larvae, the post larvae love that they have their homing, and they love to stay at the same place. So what I noticed is that when they are on the coralline algae, they tend not to move to another part of the plate, although there would be diatom on another part of the plate where there is no crystals coralline algae. They don't like to move. They move after a while, then they start to move. But in the first stages, they tend not to move. They like to be at the place where they've settled. So if the diatoms cannot grow on the coralline algae, then at the end, they end up staying really weak or really small. But ideally, it would be nice, but they would have to move.
Maybe the new trend for the research will be to teach them where to go to eat.
Yes, you're in charge.
Okay.
About the diatoms, I think that we must be careful, because maybe the diatoms are tricky. Because probably it happens in abalone, maybe also happens in limpets, that each species has the specificities for the type kind of diatoms, probably. So maybe you can place a diatom and it can be an inhibitor of the species. So you need to check the species before to see if it can induce the settlement. Otherwise, you can place a diatom species that is not good for this culture species and will inhibit the settlement. So then diatoms can be tricky. In an experiment, we see that Aspera didn't like Amphora, didn't like Navicula, and Canday only like Navicula. So,
Sure.
Very well. Now another very precise question to Gercende from Carla. At what time post-fertilization do you transfer the hatched larvae to the larval tanks?
The hatched larvae to the larval tank. As soon as they are hatching, as soon as we observe the larvae have hatched, they are transferred to the larval tanks.
Okay, you just don't wait. It's just,
no.
Very well. Okay. Now, Gercende is asking to Diego, what spawning induction methods have you been tested for limpets. Diego.
Well, Mirko already exposed this thing much better than me about, different thermal shocks, bubbling, ultraviolet seawater, peroxide nitrogen, the most typical ones.
Okay.
Maybe some out of today also high concentration of algae.
That's why you have to speak with each other.
Yeah. Thank you. Mirko gave a good explanation of the different induction methods, and I was amazed by the results you could get for the induction with limpets. Great work. Yeah,
yeah, we will see this winter if we are able to repeat the same experience. This will give us more amount of data and to be sure which kind of method we can use for the induction. Of course, I saw a question I didn't explain during my presentation, but I saw the question from Diego. After these experiments, we check some of the specimens which were not spawning, just to know which was the stages of the gonads, and they were mature. So now we have also to think about how to play with this treatment. Maybe more intense for a longer time, different concentrations in the case of hydrogen peroxide, different power in the case of UV light, but even if this method wasn't successful, we still think that can be used for the induction.
Okay, Carla is still precising. She wants very precise things. Gercende, what you consider old lands, how many days? That means, how do you distinguish older or young from the old? That's the point. When do you think that they got old? They are not young anymore.
Yeah. What we've tried until now is what we considered old in the experiment. They were 45 days old, Olvella lens, the germlings of 45 days old. And the young ones were like 4 days old, only after the release of the germlings. And normally, for the normal induction in the tanks, we are leaving the germlings between 15 days and 3 weeks on the plates before using them for settlement induction, more or less.
Mm-hmm. Very well. I think it was quite good. Carla is satisfied. She's putting the... Diego is asking you what glue is used for the tags. Mirko also can give what he's using, but Gercende, you have answered, Gercende. You have just you have answered this question, right? What was your question? What glue do you use?
We normally use, I mean, the one that we found that works well is the 2-component epoxy that is drying in 5 minutes, more or less, and it's quite stable. We've tried the super glue also, but the super glue tends to break after a while. It's not elastic. It's not lasting for that long. What did you use, Mirko, for yours?
Super glue, I think.
Yeah, super glue.
Okay. Well, at the end, the ones that lasted the longest and that remained the longest on the shell, because they were kind of elastic, were 2-component epoxy that you buy in any DIY shop, and they tend to remain for quite a long time, and they don't drop.
Nice.
Yeah,
Diego is asking Mirko about the maturity stage of the gonads from the specimens used in spawning, if it was checked after the trial.
Yeah, I answered this question before.
Yeah,
so that's yes, they were mature, and this is the reason because the future research will be orientated towards the intensity of the stimulus and yeah, basically the intensity of the stimulus.
And what rate of spawning adults was obtained, Mirko?
Sorry?
Diego is asking what rate of spawning adults was obtained.
Well, this was once trial, as I told you before, and we got the main result were in the bubbling treatment, in which we get of 4 males, we get 3 males spawning. Then in the temperature, we get both females and both female and male, while in the peroxide we have only male. The same treatment of hydrogen peroxide, we get just 1 male in Patella aspera.
But in percentage, more or less, what is 50?
Okay, percentage in the bubbling method is 35%, considering all the numbers, and the other are, let's say, 2 on 80 and 1 on 80.
Bueno, está bien, está muy bien, muy alto.
Yeah. Yes, it means 2 on 80 is 25%, yeah, with the temperature, and 1 on 8, yeah. Okay.
No, it is high.
That's the math.
Oh, congrats.
Okay.
Yeah, I'm sure that the sensation we have during the experiment is that really depends from the stage of the maturity stage of the limpets, and of course it is something that we cannot control, but maybe next research and we can discuss it. But I think it can be the use of gonadotropin analog to try to see if we can get at the same time the maturity stage, which is ideal for the induction of the release of sperm and eggs.
Oh, see, yeah, it looks like interesting. Very good, very good, very good result. 20%, wow. Congrats.
It's getting exciting, right, Diego?
Yeah, 20% in the thermal shock. In the bubbling shock is 30, 37, 35. So it's 3 on 8. Yeah.
And the trial of the gonadotropin will be very interesting because you can obtain ortegon synthesized state very well developed. And maybe you also can use the gonadotropin to induce the spawning also, not only to test, you know, the methods.
Yeah, sorry, I don't understand.
The gonadotropin also can be very interesting to test for the induce the simultaneous maturation of all the specimens, but as well as induction itself for only the gonadotropin with a great quantity.
Yes, that's something else that can be tested for sure and can work.
Yeah, maybe according to the biology, it can be possible a very high ratio of spawning specimen.
Yeah, we have to take care when the injection, because we saw that it works with to get the gametes or to check the sex of the individuals, but you can have mortality can be high, especially with Patella aspera.
Yeah.
So it's called non-lethal injection, but
the
percentage of casualties can be high.
Okay, very well. We have a last question to Gercende, which Carla is asking what temperature, at what temperature you maintain the gametes before the fertilization in Abalone.
We maintain them at the normal seawater temperature that we are doing while we are doing the spawning. I mean, here in the Canary, the temperature varies from 18 to 24. So we are performing spawning throughout the year. So it depends at the time of the spawning what is the temperature of the water, but they are kept at the normal natural seawater temperature.
Mm-hmm. Very well. So, and this was the last one.
And we've got a direct message from Carlos. I don't know. He says, Haliotis culture is well established. What lesson can we learn from it regarding the work to be done with other invertebrates, particularly limpets? Well, to answer Carlos, I think that that's what we are doing in AquaInvert, trying to learn from what some of us have found in one species and try to see if it works for another one. I think here you've touched the essence of why the AquaInvert was there.
And what is the similarity on the invertebrates?
Because we are finding a lot of similarities between. I mean, there are dissimilarities between them, but we are finding a lot of similarities, especially for settlements. The more we talk about it, the more we realize that so many things are common.
And not only between limpets and abalone, but also with. Seems also with sea urchin. So
yeah. Also, the echinoderms seem to have many common things also.
Yeah, and we know that.
And then Carla was asking, do you see differences in fertilization with temperature? No, we've got two main. We have two moments during the year when we see. That the percentage of animals is higher, the ones that are mature, and it's when there is the temperature increase and when there is the temperature drop during the year. These are the moments when we see in the number of broodstock that we have a higher percentage that are mature, but normally throughout the year we always have a percentage of the population of the broodstock that is mature, and that allows us to induce spawning. And we don't find differences in fertilization with temperature.
Can you hear me well or no?
Carla, não conseguimos ouvir.
We cannot hear you, Carla. It seems that you are in a submarine or something like this.
Yes. Okay, Carla. You have the chance of meeting almost us all in Madeira next week. Keep it with you, the questions and what you want to say. Okay. So it was a very interesting webinar. I think that we have arrived. We are getting new results, a new platform for more discussions. It's quite encouraging, all this that we have been talking about, and very exciting in some points. So I hope to go on also hearing from those that are not involved in the project. Please keep in touch. For the next webinars. The next one will be on the 30th of November. So don't forget, the next one will be probably even more exciting than this one. And I wish you a very good weekend, which is nearby, and hope to see you all soon. Okay. Bye. Thank you.
Thank you.
See you.
Goodbye.
Ciao.
Goodbye. Thank you to everyone.